Hello students, in this video I am going to tell you about the process of oogenesis. As in earlier video we have seen the process of spermatogenesis and the hormonal control of spermatogenesis. In this video we will discuss about the oogenesis. So first thing, what do you mean by the process oogenesis? So it is the process of formation of functional haploid ova from the diploid germinal cells in the ovary, or simply we can say it is the formation of process of formation of ova from the ovary. But this definition is more appropriate. It is more appropriate. Why? Because there is a term functional haploid. I am emphasizing this term functional because you should know that at the early stages of oogenesis occur. Do you know when? By the time when female fetus is 25 weeks old. So do you understand this, that oogenesis occurs during female fetus only when it is only 25 weeks old, and at this time all the oogonia have been formed, and no oogonia added after the birth of the female child. Point to be remember, all the oogonia have been formed during this stage only, and none of the oogonia is added after the childbirth. So it has been formed and entered into primary oocyte stage. So once again, I'm explaining you that it is the formation of functional haploid ova from the diploid germinal cells in the ovary, or you can say female gonad. And at early stages of oogenesis occur by the time when female fetus is 25 weeks old. Point to be remember, and all the oogonia have been formed and entered into primary oocyte stage. So this process also occurs like multiplication phase, growth phase, and maturation phase. So simply we can see that oogonia and it goes to mitosis, differentiation, and form primary oocyte, and this will goes to first meiosis or meiotic division, and this first meiotic division is completed prior to ovulation, and they form secondary oocyte, and this will form an ovum. So it looks very simple that oogonia after mitosis differentiation will form primary oocyte, then it goes for first meiotic division and form secondary oocyte, and this secondary oocyte ruptures into an ovum. But this is not as simple as you can see this. So let's come into the detail of this process. So first thing, that first process is called multiplication, when certain cells of germinal epithelium of ovary which are larger than others function as germ cells, and it undergoes repeated mitotic division and it form diploid oogonia. So the very first step, as I shown you, that oogonia is formed. So oogonia is formed by the process of repeated mitotic division and then it start the growth phase. Oogonium grows in size and function as primary oocyte, and there is a covering around primary oocyte, and this in-sheathed primary oocyte is called primary follicle. You can see here in this structure, this is a section of ovary. This outer region is cortex, inner region is medulla. These are the blood vessels, arteries and veins. This is the primary follicle. You can see here primary follicle. Then primary follicle degenerate between birth and puberty, and at puberty it contains about 60,000 to 80,000 primary follicle. Point to be remember, at puberty it contains 60,000 to 80,000 primary follicle, but in the fetal stage it contains more than that. But at puberty it remains 60,000 to 80,000 primary follicle. So in the maturation phase, what will happen, that primary oocyte begin meiosis first, but the division is arrested in dikinesis stage of prophase first. Point to be remember. This division is arrested in dikinesis stage of prophase first of meiosis. It remains till the female attains puberty. So primary oocyte remain in meiosis first until the female attains puberty, or you can say at the stage of now this puberty stage is decreased to 11 years to 13 years. So they have to wait for this, and it get arrested in the meiosis first or prophase first at the dikinesis stage. And what will happen at puberty? At puberty, follicular sheath differentiates into layers of granulosa cells, and another layer called the theca developed from cortex. You can see in this diagram, this is the cortical region and there is theca developed, and so many layers are gathered around. So, with the formation of theca, primary follicle transferred into secondary follicle. You can see the structure. This is the secondary follicle, and you can see here that there are so many insheathed structure around the this, and this is called as secondary follicle. And after the secondary follicle, granulosa cells secrete fluid, and that cause the development of an a cavity or antrum. You can see here, this is the antrum around the primary oocyte, and this stage is called, see here, this is the antrum, this is mature secondary oocyte, and then it form tertiary follicle, and this is the you can say it's a hollow space, and this is called as an antrum. So primary oocyte grows further and complete meiosis first. This primary oocyte grows further and complete meiosis first, and secondary oocytes which contain small cytoplasm and the whole number of chromosome set, it will form this primary oocyte. After completing the meiosis first, will form secondary oocytes and the first polar body. This primary oocytes, after completing the meiosis phase first, as I told you, it was arrested in meiosis first at the dikinesis stage, but at the time of puberty it complete its meiosis first and form secondary oocytes and polar body, and this follicle grows to its maximum size and form Graafian follicle. So you can see here, this is the Graafian follicle. Now this secondary oocytes grows its maximum size, and you can say Graafian follicle is the last step in the formation of follicular stage, and they form a Graafian follicle. This is the you can say this Graafian follicle is the matured stage of follicular stage. So follicle grows to its maximum size and form Graafian follicle. Zona pellucida develops around the secondary oocytes. This layer is called zona pellucida. See here, this layer is called zona pellucida. Now the secondary oocyte goes for meiosis second, and it get arrested in metaphase second due to accumulation of MPF, that is metaphase promoting factor. Point to be remember that again now primary oocyte were arrested in meiosis first, and this will arrested in meiosis second uh, due to the accumulation of metaphase promoting factor, and uh, ovum is shed during ovulation. So in this stage, ovum is shed during ovulation, and then ovum goes to oviduct, that is fallopian tube, where at the ampulla part, ampulla isthmus part, cell cycle resume, and only after the entry of sperm Cell cycle will resume when sperms reach to at this point, that is fallopian tube, that is called as site of fertilization. Actually, the site of fertilization is ampullary isthmic junction in fallopian tube. So once the sperm entered at this place, the cell cycle resume again, which was arrested in meiosis second. It triggers breakdown of this MPF, that is metaphase promoting factor, and promotes synthesis of APase, that is anaphase promoting complex. So meiosis second is completed and second polar body has formed, and this will form oocyte. Oocyte will differentiate into an ovum, and sperm and ovum meets and they will form zygote, and zygote will develop into an embryo. So you once again see this structure: primary follicle, secondary follicle, Graafian follicle. This is the corpus luteum. It ruptured to form this structure, Graafian follicle, and also releases progesterone. And this is tertiary follicle. This region is medulla, and this one is the outer one is the cortex. And so you can see in this structure also how it goes. See, this is the fetal life. Oogonia has been formed, which develop into primary oocyte at birth, and it remained in this stage only, arrested in metaphase first meiotic division completed prior to ovulation. So this is childhood. Then at the puberty, meiosis occur and it form secondary oocyte with first polar body. And in adult reproductive life, once the sperm reach to fallopian tube, then meiosis second occur and they will form the secondary oocyte and second polar body, and then they get this secondary oocyte, they get differentiated into an ovum, and this ovum uh, conjugate with fuse with sperm and form zygote, and this will form an embryo. So this is all about the oogenesis in case of human female reproductive system.